My name is Kristin Knouse, and I'm a graduate student in Amon's Lab at the Koch Institute. The Amon Lab is broadly interested in the mechanisms by which cells progress through the cell cycle and segregate their chromosomes, and the consequences that arise when these processes do not occur properly. So as a cell progresses through its cell cycle, it has to achieve certain things at specific times in order to accurately replicate its genome and make two daughter cells that are identical copies of each other and have the exact same genome. And the way they do that is they first progress through a phase called S phase, where they replicate all of their DNA. So now every chromosome is duplicated to form two sister chromatids. They then have to pull those apart into two daughter cells, and they do that during mitosis. During mitosis, an organelle called the centrosome will start to nucleate microtubules. Microtubules are these long filaments that form a spindle. They will attach from the centrosome to the chromatids, and they're able to pull on the chromatids to put them in the right place. And that process generates two new cells with identical genomes. Cancer cells are notable for having very high levels of genomic instability. First being genomic instability at a gene level with high levels of mutation in various genes, and also at a chromosome level with varying numbers of chromosomes. So they do have at least two copies of their genome, and in many cases along with that will have multiple centrosomes. And in those cases it has been shown that having multiple centrosomes and multipolar spindles allows for very high levels of chromosome segregation, and that contributes to the overall genomic instability that characterizes cancer. What is peculiar to us is that the liver has those features of cancer. It has multiple copies of its genome and it has multiple centrosomes. Yet it's pretty obvious that livers in most people are not immediately transitioning to cancer. And so we want to understand, how is the liver different from cancer? What mechanisms does it employ such that it can, even though it has multiple centrosomes, segregate chromosomes accurately, or accurately enough to maintain organ function and not become a tumor? And so what you see in this image is a cell trying to segregate its chromosomes, but instead of having two spindle pulls, there are at least three, perhaps unseen to us, four or more. My research is trying to reconcile this paradox of how a hepatocyte, being polyploid, having multiple copies of its genome and also multiple copies of its centrosome, can undergo what appear to be robust and functional cell divisions. And so that enables the cell to form a perfect bipolar spindle and accurately segregate the chromosomes into two daughter cells. I find this research very interesting because ultimately it's characterizing a fundamental aspect of human biology. At the same time though, some of the characteristics of hepatocytes are shared with cancer. The fact that they have extra copies of their genome and extra centrosomes. And so I think that in addition to really understanding a very fundamental principle of how this organ works, it may also lend insight into how cancer has a means of making these processes not work. And in uncovering those differences, we might now reveal new therapeutic targets for the disease.